This video is for educational and informational purposes only. The content is based on personal experiences and is not medical advice. Always consult a qualified healthcare professional for personalized medical guidance.
Depends on who you ask. It's not universal in the response to our stuff. If you ask oncologists who take care of folks every day, they say it's all nonsense because they never heard about it. If you ask scientists, they say, yeah, well, we know mitochondria have problems, but we still think it's a genetic disease. Otherwise, I won't get my grant from the National Institute of Health. When you talk to folks, they seem to want to know more about this. I haven't spoken to people in the pharmaceutical industry. I don't know what they think. They more or less do whatever the boss tells them to do. I find that's mostly what happens. My colleagues think they're locked into confirmation bias, and I think a lot of folks are that you can't buck a system that's been in place for so many decades, even if you have accurate science. So I don't know how you respond to that. It can't be right because everybody else thinks differently. So I don't think anybody wants to have the apple cart turned over. I think everybody wants to keep the status quo, regardless of what field or occupation or discipline you're in. I think the people, of course, would like something different. Brain cancer, all cancers. I get hundreds of emails from everybody, every type of a cancer, and they all want metabolic therapy, but the institution, the system, is not allowing it for a variety of reasons, and none of it has to do with patient health. It hasn't. There's been no progress in 100 years, not 20 years. If you go back and look at the data from Bailey and Cushing in 1926, they said that people with glioblastoma and very aggressive brain tumors would survive anywhere from 8 to 14 months, which is today it's about 8 to 16 months. So that's you got an extra couple, 3 months in 100 years. So you're right. It is, but it doesn't have to be. It doesn't have to be. This is the great tragedy. We can manage that. We think we can make 4 years the average survival rather than 15 or 16 months, maybe even longer. I wrote a paper in 2010 in Lancet Oncology. We were just so upset about the tragic loss of lives with this disease, GBM, of course, that I just fired it out and I said, let them know on that. And we clearly said that the standard of care is responsible for the demise of these people. Because as a neurochemist, I understand the glutamine-glutamate cycle. And we know in the brain, glutamine is a very necessary amino acid for the synthesis of glutamate, which is an excitatory neurotransmitter. And it has to be removed very quickly after being released, that is the glutamate, because it impacts on NMDA receptors in the neighboring neuron to transmit signals from one cell to the next. And if it's not removed quickly, it can lead to excitatory seizures and excitotoxic death of cells. So we knew this, and we knew that as soon as the glutamate is released into the synaptic cleft, the glial cells, which embrace the synaptic cleft, would take up glutamate very rapidly and metabolize the glutamate to glutamine, which is not excitatory. And the glutamine would then be released by the glial cell, and taken up by the neuron. And the neuron would then metabolize the glutamine back into glutamate and consolidation in synaptic vesicles. And this has been studied in the neurochemistry field for decades. It's called the glutamine-glutamate cycle, and it protects the brain and neurons from excitotoxic death. There had been some reports that in glioblastoma, there was a significant amount of extracellular glutamate, which was leading to excitotoxic death of neurons just in the microenvironment. And I knew that there's plenty of astrocytes that would be sucking this up. But then we also realized that the tumor cells themselves depend heavily on glutamine. So if the glutamate is being released from the neuron, or the neuron dies and a significant amount of glutamate is now in the microenvironment, the astrocytes will suck up this glutamate and then dump it out as glutamine. And if there are cancer cells in the microenvironment, they're going to be sucking down the glutamine. And we now, at that time, in 2010, we knew that debulking surgery followed by radiation. Radiation heats the brain and would lead to significant neuronal death, more glutamine cycle disrupted. Any surviving tumor cells in the microenvironment would be bathed in a fuel that they need to survive. And not only that, after your brain is irradiated, and right after surgery, and even before surgery sometimes, because the necrosis and inflammation and edema in the tumor microenvironment is treated by high-dose steroids. So we knew that the steroids would raise blood sugar levels. And not only that, when you irradiate somebody's brain, blood sugar goes through the roof. It's an attack on the body. So we have two fuels that the tumor cells absolutely have to have. The sugar, glucose, created by the radiation and the steroids. And then we have the glutamine being dumped out onto the microenvironment, leading to the survival of the tumor cells, making them massively resistant to any further attack by chemo or radiation. Because the waste products of glucose and glutamine, which we now know is lactic acid and succinic acid, protect the tumor cells from any further attack. So the reason why they recur and are resistant, because of the very treatments that these poor folks are receiving by the standard of care. It makes no damn sense. If you have any clue about neurochemistry, neurobiology, or cancer biology, what you're doing to your patients is essentially depriving them of any possibility for long-term survival. I have published this over and over again, detailing all of this, and it falls on deaf ears because the system is so profitable using radiation and these crazy things that it's not possible to shift gear regardless of what the patient, how it might affect the patient. This is immoral, absolutely immoral and ignorant on the part of the field. So you take patients and you don't do that, and they live longer. And they can live a hell of a lot longer if you didn't irradiate. And those paper you're talking about, because my Greek colleagues were still forced to do that stupid radiation. And they know if we didn't irradiate those people, we can even get them to live far longer. Well, the system locks in, it prevents alternative views to a standard of care. So they'll say, like at Cedars-Sinai in Los Angeles, you can treat without standard. You can treat with standard of care, but you can't do metabolic therapy unless you've already done standard of care. So you've already stacked the deck against the patient. So what are you going to do about that? Oh, it's still not readily available. Again, this was done before some of those patients were being treated years ago, and we now and it was before the big treatment protocol that we just wrote at the end of 2024. We've written a very comprehensive treatment protocol, which says don't irradiate the brain, basically. So, and we gave many options of how you negotiate the treatment strategy depending on the individual. One of the authors on our papers is Dr. Joseph Maroon, former president of the Neurosurgical Society of America. And he told me he operated on 400 GBM patients on his life, not one survived. So he said, we got to do something. So, we know. And I gave a lecture to Henry Ford Cancer Center, to the radiation oncologists, their whole division of radiation oncology. How do you think that went over when I told them you were killing all your patients? That didn't go over well. You might as well have been, all I did was get glares, rather than saying, well, I guess it was pretty harsh to tell somebody that their whole life is just like, what are you doing? Remarkable. You'd think they would be excited about it. But they're threatened, or feel threatened. And it's really a very unusual situation. But it's not, when you go back and look at massive paradigm changes from the past, you find the same kind of reaction by the establishment. This is what I'm experiencing here is no different than what Galileo had to deal with, and Giordano Bruno and Kepler and these other guys when they were defining the sun as the center of the solar system. I mean, this threatened the supremacy of religious organizations and things like this. So, and it takes decades sometimes to accept even what is obvious. And we still have 15% of the American public still thinks the sun revolves around the earth. So we've made a lot of advance, but not complete. You can say the same thing for Louis Pasteur. He was brutalized, even though he came up with a rabies vaccine and found out that it was germs that was causing infectious diseases. And the French hierarchy belittled him. And we still have the same problem with the Darwin-Wallace theory of evolution, which you cannot understand glioblastoma or any cancer effectively unless you understand evolutionary biology. It's not possible. So if you have people that don't understand evolutionary biology, they're the same people that would put the alligator stuffed with herbs in your face. And not only that, they get very well paid for this. I mean, you're talking about these radiation oncologists get a lot of money. So now I'm not saying all radiation is bad. I think for brain, it might be for sure. But I think for as a final removal of some lesion, a proton beam could be the solution to the problem. But if we take a glioblastoma and shrink it down and make it less angry, less inflamed, less angiogenic, where now the surgeon can debulk a far greater amount, and what's remaining may be susceptible to a low-dose chemo or even these parasite medications that we have. But we would not, and we do not recommend radiation to the brain for glioblastoma or any brain cancer patient for that matter. But that does not mean we could not use radiation for lung cancer, colon cancer, or something like that. If the lesion remaining is tiny and compacted, because if you don't do that, you could run the risk of spreading it all over the place. And don't forget the reason why radiation, it kills through reactive oxygen. Reactive oxygen is like a bomb, a local bomb blows up, kills the cell. But if you don't kill that cell, and that cell is fermenting glucose and glutamine, it's got a kind of an acidified shield around it, making some of these treatments ineffective for killing off all the tumor cells. So you really have to put the tumor, regardless of where it is, because what we have found is that all tumors, regardless of where they're localized, have the same metabolic problem. They are powerfully fermenting glucose and glutamine, dumping out large amounts of lactic acid and succinic acid, and cannot shift to ketone bodies or fatty acids to replace glucose and glutamine. So what I just told you is the solution to the cancer problem in a nutshell. But that's not taught in any medical school. It's not, other than our papers, there's no one else saying this. Yet we have all the science to support what I'm saying. Based on historical observations from Otto Warburg, we have a big paper that'll be coming out, just accepted for publication in Bioenergetics and Biomembranes, which really tells the entire cancer field where we went wrong and how to get back on track to manage cancer effectively, because it is a mitochondrial metabolic disease. So the mitochondrial metabolic theory will replace the somatic mutation theory, and then the field can start moving forward with treatments that are far less toxic and far more therapeutic, because it's based on the science as we know it. So the cancer cell is simply falling back on its default energetic state, which is fermentation. All organisms on our planet began in an anoxic environment. There was no oxygen in the environment. So all the cells, the living cells at that time, were fermenters. There was no oxygen. They were all single cell fermenters, and they would grow, divide, and would live as long as there was an availability of fermentable fuels in the microenvironment. Then we had certain archaeobacteria that were able to produce oxygen. And over time, we developed oxygenated environment. It was the oxygenated environment that allowed us to form metazoans, which are multicellular organisms, because the fusion of the archaea with other fermenting organisms at that time now produced a hybrid system where one organism could use oxygen as an acceptor of electrons, and the other organism was linked to a fermentation metabolism. And then now we have division of labor, and we have energy efficiency, which led to metazoans, which are multicellular organisms. Now we know from the work of Sonnen Shinisato, Carlos and Anna down here at Tufts Medical School, they have been professing for a long time that the proliferation is the default state of all metazoan cells. When no longer held in check, they will naturally fall back on proliferation. And what we have shown is that the default energetic state is fermentation. So what's keeping cells quiescent and differentiated is the action of the mitochondria, producing highly energy with using oxygen as an acceptor of electrons, leading to a highly energetic state, allowing cells to differentiate and form different organs and different systems, and can operate under different conditions. Well, the mitochondria energy gradient inside that organelle, which has its own DNA, as a matter of fact, relinquished all genes to the nucleus except 13 genes as part of the electron transport chain. So they essentially control the destiny of the cell. And when the cell gets sick, they release cytochrome C and the cell dies to protect the rest of the cells from any dysregulated cell growth. So the organelle that controls the destiny of our existence is called the respiratory enzyme. This is what Otto Warburg got the Nobel Prize for. He found out the protein that actually allowed cells to use oxygen, which is cytochrome C. So when you take cyanide, you block cytochrome C's ability to move protons, and you kill the organism real quick. So when cytochrome C is released from the mitochondrion, it's a signal to die. So you're not respiring anymore, and that's called a program cell death, apoptosis, program cell death, which means the mitochondria are damaged or inefficient in some way. Cytochrome C is released, sending off a signal to die. That's called program cell death. An organization, when you're not able to function energetically any longer, there's a program that triggers your death. And basically, when oxidative phosphorylation is chronically disrupted over time, which was Albert Szent-Györgyi oncogenic paradox. He wrote that in 1977 in the journal PNAS. And every breath that we take, that's why we're breathing here. Every breath that we take brings oxygen into the system. Every exhale comes out, it's got CO2 and water moisture. So that's the waste product. The waste product of oxidative phosphorylation is CO2 and water. And the CO2 are the carbons from the foods that we ate, and these foods are broken down systematically in the Krebs cycle, and then we generate these reducing equivalents, which deliver their electrons to the proteins of the electron transport chain, which is the mitochondria, and the proteins are moved from one complex to the next, from one protein, and they pump protons into this. The electrons are passed from protein complex to protein complex that's embedded in the inner membrane of the mitochondria, okay? And there's lipids, and these lipids in the inner membrane are facilitators of how efficiently this whole thing will work, like cardiolipin, a unique lipid in the inner membrane. And the electrons are passed from one complex to another, and these complexes are multi-dimensional protein conglomerates. And in the process of moving the electrons, the protons are pumped from the matrix into this inner membrane to space to form a gradient, a high proton enriched gradient, which then pass through another protein complex called the F1 F0 ATPase, and that spins like an impeller. And you're right, there's transporters that bring ADP into these mitochondria, and the phosphate is then added to ADP to form ATP, which is so highly efficient with the waste products of CO2 and water. So most of the energy in our cells is coming from this oxidative phosphorylation. Okay, so in cancer, that process becomes gradually corrupted, not acutely, because if you acutely disrupt OXPHOS, you kill the cell. And the example of that is cyanide. Look how fast you will die. That's called rapidly acute death, because you block cytochrome C, you block the movement of the electrons through the complex, and you block the ability of oxygen to be reduced to water. So this is basically how fast you can die. Now that does not happen in cancer. It's a gradual process. And when I go back to the oncogenic paradox of Albert Szent-Györgyi, he said something unique about cancer. It can be caused by almost anything we can think of, whether it's a virus, an inflammation, a radiation, a chemical carcinogen, intermittent hypoxia, viruses. He says, it seems like anything and everything and anything has the potential to cause cancer. But he was unfamiliar with the common pathophysiological mechanism which allowed a cell to lose its growth control and become dysregulated in growth, which is cancer. Cancer is growth control, is dysregulated cell growth or cell division out of control. So what is responsible for the control? Why cells don't do this? And the answer is because the mitochondria control the cell cycle. Through the proton motive gradient, they are controllers of the cyclins. They determine when a cell needs to divide or not divide. So they are the determiners of cell existence and destiny. Hybrid system, and my colleague, Dr. Christos Shanopoulos from Semmelweis University, is a world leader on mitochondrial substrate level phosphorylation, which we can get into. But he has that same video with, it's like a spaghetti network. It's like a bunch of worms in the cytoplasm. They are very active. The nucleus sits there as some, dormant thing, but it's the movement and the activity of the mitochondria. They fuse, they come together, or they break apart. They have their own DNA, but the DNA is in these little circles, and they have numbers of these different kinds of circles of DNA. They're a very active, dynamic organelle that leads to the entire energy balance and destiny of the cell. Ultimately, it's responsible to the second law of thermodynamics, which is disorder. When mitochondria become corrupted over age, they become gradually more and more less capable of maintaining energy balance, and you die. So you die because your mitochondria no longer are able to maintain a current across the membrane. So, and that goes back to the membrane. What's this current? Because you can't live if you don't have a current. It's called equilibrium, and equilibrium is death. So you need energy to prevent equilibrium. And every cell has a different electrochemical gradient across the membrane. For example, the heart has about a minus 88 or something like that, which means because the heart muscle is pumping. In order to do that, it has to have a very powerful sodium potassium ATPase and calcium ATPases, ATPase, breaking down ATP. You contrast that with an erythrocyte, which is a blood cell that carries oxygen around our body, picks up oxygen from the lung, delivers it to the tissues, picks up the CO2 from the tissues, delivers it back to the lung. Okay, that has a electrochemical gradient of minus 6 millivolts compared to the minus 88 millivolts of the cardiac. So interestingly enough, because the erythrocyte has no mitochondria and no nucleus, its job is simply to transport gases through the body, and eventually it's recycled. But it still has an electrochemical gradient, a very low electrochemical gradient, and the energy for that organelle, for that cell, comes purely from fermentation, because there is no oxidative phosphorylation. So they have a small fermentation, and they only need to produce a slight gradient, enough to remove gases. Whereas cardiac myocytes and neurons, which have a very powerful role, have very steep electrochemical gradients, and therefore they have to have a lot more energy, and that energy is delivered by oxidative phosphorylation. Neurons and cardiomyocytes rarely, if ever, become cancer because they can't shift to a fermentation metabolism. So that's why you can know why these neurons never form cancer, or not never, but rarely, if ever. Cardiac myocytes, only cells that have the capability of transitioning from from oxidative phosphorylation to a fermentation metabolism have the ability to form dysregulated cell growth, all controlled by the mitochondria. So people need to appreciate more this importance of this organelle. Yet the whole cancer field was based on somatic mutations in the nucleus. So the focus of the field was on something other than the important issue, which was mitochondrial biology. Yes, every one of those different injuries has a way to chronically disrupt oxidative phosphorylation, leading to what we call substrate level phosphorylation, which is the evolutionary conserved way that we had got energy before oxygen came onto the atmosphere 2.5 billion years ago. This was all single cell, all about fermentation. And the cells would continue to divide as long as they had fermentable fuels. Cancer cells are doing the exact same thing. The very organelle that's maintaining differentiated state and energy efficiency has lost that. So the kill switch, even though they can release cytochrome C, they're already locked into a fermentation metabolism, and they bypass programmed cell death, and they continue to grow in a dysregulated way, as long as they have the fermentable fuels in the environment to do that. Once you remove those fermentable fuels, these guys can't survive. And we interrogated them with all the different, we asked, whatever amino acids, how many other sugar, what keep these alive? Our big paper just showed this. We interrogated all 20 amino acids. We interrogated everything and just asked, what keeps these cells alive? And we were able to find it's glucose and glutamine, and they're powerfully synergistic. So when they get together, they will drive a dysregulated cell growth through a fermentation metabolism involving substrate level phosphorylation. Another big thing we found is that the damn mitochondria can ferment. There are fermenters too. Wow. There's a fermentation mechanism inside the mitochondria. It's called mitochondrial substrate level phosphorylation, driven by the amino acid glutamine. Glutamine is fermented inside the mitochondria. This is big. This is what was keeping everybody from understanding what the hell was going on in these cancer cells. It's unbelievable. They don't have a nucleus. You got to have a nucleus to divide. So if you don't have a nucleus, they're going to divide. So they don't have a mitochondria or a nucleus, and they can't divide. You have to have a cell that can divide. And what controls the division and regulation is the mitochondria. So a cell that can divide will proliferate like crazy if it doesn't have a regulatory system to keep it in a stable state, and that's the mitochondria. That's why it's falling back on the default state, which is proliferation driven by a substrate level phosphorylation. And you prove that. Pull the plug on the glucose and glutamine and watch them die. I want you to repeat after me. There is no diet that can target glutamine. Can you guys say that? We can easily control glucose in our environment. It's a non-essential fuel. Our body can make it under restricted conditions through the process of gluconeogenesis. But the glutamine is an essential. They call it a non-essential amino acid because it can be produced from glucose by a glutamine synthetase reaction. The problem is, for cancer, it's absolutely essential. And for our immune system, it's essential. And for our gut, it's essential. Urea cycle, it's needed. So you have an amino acid that's absolutely essential for our normal physiological functions. And it's the highest amino acid in our bloodstream. It's the most abundant. It's about 500 micromolar, 0.5 millimolar, and it's higher than all the other amino acids. And it's therefore an essential reason for the gut, for the immune system. If we get attacked by bacteria or a burn or something that lets bacteria into our body, our immune cells use glutamine as a fuel to kill bacteria and to perform its normal function. So do our gut needs glutamine, gut health. So sometimes you see people that are irradiated for cancer get high dose glutamine to repair their gut from being brutalized. So you have an amino acid that the cancer cell absolutely needs, and our immune system absolutely needs. So you have the same fuel that these tumor cells need to grow, and our immune system needs to kill the tumor cells, and they're both going to need the same fuel. That's why I developed the press pulse therapeutic strategy, where we press glucose down tight and exercise, because the exercise will pull the glucose out of the bloodstream. Our brain can switch to ketone bodies, but it will take what little glucose might be there. It leaves not much for the cancer cell. So now you come in with your strategic targeting of glutamine, but you do it in a strategic way. You don't do it in a non-strategic way. You have to know that your immune system is going to be needed. You've got to maintain gut health. You have to maintain immune system health. So, but here's the interesting thing. When you take away glutamine from the immune system, it doesn't kill them. It just paralyzes them. They can't perform their normal function. But when you take the glutamine away from the tumor cell, it kills them as long as you put the noose around the glucose. So it's a balanced system. So what we do is we put the noose around glucose, and then we come in with a pulse of a glutamine inhibitor like 6-deoxynorleucine, DON, paralyzes the immune system but slaughters these tumor cells. Then you pull the drug out. You let the patient recover. The immune system then immediately becomes active because they weren't killed, they were just paralyzed. So they come in and start picking up the dead corpses from the tumor cells and cleaning up the environment and protecting the gut. And then you come in and pulse glutamine, just glutamine targeting, just while you choke hold the glucose. And then the patient's tumor gradually gets degraded. And you don't suffer from hair loss and all the diarrhea, fatigue, and all this crazy shit that people do. Once you understand evolutionary biology and you understand the biology of the tumor, this is far less complicated than people have made it out to be. The problem, what I'm telling you, is unknown to the majority of people working in the cancer field. Even though we publish these papers in open access, peer-reviewed journals over and over and over and over again, they don't read it. We wrote the big treatment protocol we had 30 authors on this, MDs, PhDs, nutritionists, biochemists, physicians, the whole group. They set the standard. This will be the standard of care for cancer in the future. It should be today. We can save so many lives. We have 1,700 people a day in the United States dying from cancer. That's 70 people per hour. And these people are usually dying horrific deaths. They're not surviving the toxic treatments that they're given to these folks. Whether you have a brain tumor, colon cancer, lung cancer, breast tumor, they keep poisoning and irradiating these people using immunotherapies that sometimes work, but sometimes can kill you. And that's based. So you have to say, under what theory is your treatment being applied to me? And if they say, well, it's a genetic, some people don't even know. They don't even know what the hell theory they're working under. So if you're saying it's a genetic disease, then you're working under the somatic mutation theory. And our new paper that will be just accepted will be coming out in a few weeks. That clearly shows that the somatic mutation theory is no longer a credible theory to explain the origin or treatment of cancer. So once they come to know this, it will allow original thinkers to say, we got to go this direction. You either get on this direction or get the hell out of the way, because you're partially responsible for the death rate of these poor folks.